Hello everyone, my name is George Georgiou. I'm a curator here at the EMBL-EBI for the Gene Ontology Annotation project. I'll be talking to you today about the new web browser that we have called QuickGO, and a little bit about the gene ontology and how we do annotations and how this can be useful for your research. So to start, what is the gene ontology? Well, the gene ontology is a way to capture biological knowledge in a written and computable form. It is a set of concepts and the relationships to each other arranged as a hierarchy. So on the right-hand side of our screen, you can see that we have what we call the ancestry tree of gene ontology terms, or GO terms. It starts at the top with a very unspecific concept. So in this case, we start with biological process, and as you descend down the ancestry tree, we go to more specific concepts. These are all interconnected, as you see, with arrows of varying colors. These dictate the relationship between the terms and the gene ontology, and I will discuss this in a few slides. The gene ontology tries to capture pretty much three things. These are the aspects of it, and they're structured ontologies that can capture the biological process that a gene product may be involved in, and we refer to gene products typically as RNAs, proteins, or protein complexes. The other thing that we try to capture in the gene ontology is a molecular function. So we can say that something is involved in protein phosphorylation, and its molecular, that would be its biological process, and its molecular function would be kinase activity. And then last, we try to capture the cellular component, where the biological process and function actually happens in the cell. So in this case, following the example of protein phosphorylation, we can say that the process is protein phosphorylation, the molecular function is kinase activity, and the cellular component, in this case, we'll just assume that it's happening in the cytoplasm. And the scope of the gene ontology, or GO, is to pretty much describe the normal functions, processes, locations that gene products are involved in. What the GO does not capture, however, is pathological processes, experimental conditions, or temporal information. There are other ontologies that would capture this, but that is not our focus for today. So you're probably wondering, how can you use the gene ontology? Well, the gene ontology is mainly used to access gene product functional information. So if you have a gene of interest that you want to study, but you don't know anything about it, the gene ontology would be a good way to see if anyone's captured this information using GO. You can also use it to analyze high throughput genomic or proteomic data sets. In this case, this would be referring to, let's say you had a knockout cell line that you compare the wild type to the knockout using mass spectrometry, and you wanted to see what areas, what biological processes and molecular functions were impacted by the loss of this particular gene that you knocked out. Gene ontology can give you a nice summary of these areas. The other thing that it can be used, following along the lines of mass spectrometry per se, is the validation of experimental techniques. So if you wanted to make sure that your gene product of interest was in the right compartment of the cell that you isolated, let's say vesicles or a cytosolic fraction, the GO can be useful for actually trying to capture that. It can also give you a broad overview of a proteome. So if you want to see what knowledge is available about the entire proteome of, let's say, your favorite bacterium, you can check the GO to see what's been manually captured or electronically captured, and I'll go into these ideas of how we capture information in a moment. And then finally, obtain functional information for novel gene products. So one thing that the GO has is prediction pipelines that can actually tell you roughly, based on homology, what your protein of interest, if you know nothing about it, might be doing. Now, let's talk about the ontology structure. This is actually quite important because all the GO terms are linked by relationships, and understanding how these relationships connect to each other can help you better use the GO and interpret your data. So, as you see here on the ancestry tree again on the right, and right next to it in this little legend, you have the colors of the different arrows with the different types of relationships. And these relationships are is-a, or rather is a subtype of, part of, regulates, positively regulates, negatively regulates, has part, and occurs in. And if you'd like more information on these, in particular for you to read, you can follow the web link there. But I'll tell you briefly about them in a very generalized format. So the first relationship we'll look at is a. So we'll use the example of the urea cycle here. So we see that the urea cycle is a type of urea metabolic process and is a type of an amide biosynthetic process. Now, urea cycle is a child of these two terms, and it's directly related to them. So that's why we use the relationship is-a. Next, we have the relationship part of. For this, we'll use the case of photosynthesis with its children terms. In this case, we're now talking about the photosynthetic dark and light reactions being a part of photosynthesis. Finally, next we have the regulate terms. So we have regulates, positively regulates, and negatively regulates. And we'll put this in the idea of the immune response, where a gene product can regulate the immune response, or it can activate the immune response. So we would say positively regulates the immune response, or it can suppress the immune response, where we would say negative regulation of the immune response. Next, we have the relationship has part. So this one's a bit more interesting to grab onto. So urea cycle, the example we're using again is the urea cycle. So urea cycle has part arginosuccinate synthase activity. And in this case, the urea cycle always involves it, but if a protein has this activity, it's not necessarily taking part in the urea cycle. That's why we say it has part. And finally, the last one, occurs in. This mainly relates to cellular component, but in this case, we'll use the case of intracellular transport, where we say intracellular transport occurs, surprise, surprise, in the intracellular compartment. So how do we capture this information? How do you guys actually witness what we use the Go for? And that's the answer is through Go annotation. And what information do you need for that? Well, you need the gene or gene product identifier. So in this case, we can use a unit product identifier for a protein. We need the Go term and its ID. So for the example we give here, we give a protein serine 3-amine kinase, a reference to support the Go term, and finally an evidence code to tell us what type of evidence was offered. I'm going to go a little bit more into evidence codes in a bit. Also, and there are cases where we have two types of modifications to an annotation. The first one, and it's actually quite important for the new QuickGO, is the Go term qualifiers. And these modify the interpretation of an annotation. And the available qualifiers that we have are not, colocalizes with, and contributes to, and we also have not terms for those two. Now, what these actually mean is in the case of, let's say we use the qualifier not, we had a gene product that would most likely, we would assume has a particular function, but then it turns out it actually doesn't. So let's say a homologous protein would have kinase activity, but this particular gene product, though it is homologous to it, does not have it. So we would use the not qualifier. Co-localizes with, in this case, let's say we have a protein that co-localizes with the mitochondria, but it's not in the mitochondria. This is actually how we capture this information, by using the co-localizes with qualifier. And contributes to would refer more to protein complexes, where you particularly, let's say, have a multi-subunit protein complex that has a particular enzyme activity, but the individual components don't actually have the activity itself. It only has activity when it's in the complex. So we would then use the contributes to term. And the not terms for these two would cancel out the co-localizes with or contributes to. And last, we have the annotation extensions. These are something that are, again, is kind of important for the new QuickGO. This allows us Go curators to actually combine Go annotations with other Go terms. So normally you would have to create independent annotations for each Go term, but in this case, an annotation extension allows you to connect functions with processes and processes with other processes, or a process that only occurs in a cellular component at a given time. So, and the other thing that allows us to do is use terms from external ontologies like Uberon. So if we want to say that this particular process happens in this particular type of cell. And on top of that, we can also use database identifiers representing genes or other products. So we can say that a DNA binding protein binds to this specific gene sequence using an ensemble identifier. So to give you an example, we have in green text rather, a simple Go annotation would be PAP1 regulates transcription. But the extended Go annotation would say which genes it actually interacts with. So those are highlighted in blue. And another example we have here is TMEM115 is located in the nucleus, so that would be our simple Go term, but then we want to say the particular cell types. So it's in epithelial cells that are part of the cervix epithelium. Now I mentioned before evidence codes, and these indicate how the annotation of a particular Go term is supported. And these are based on the evidence ontology. So let's take the example for protein X here. We say that how the Go term, we're going to use the Go term alcohol dehydrogenase activity. It's up to curators like myself to find a source that provides the data that supports this term. And once we find that piece of data that supports it, we attach an evidence tag, an evidence code to it. So in this case, we're using the evidence code IDA from this particular paper, and the evidence is experimental method manual. So to give you an idea of what we use in terms of evidence codes, these are the ones that we use in Go curation that map to different ecocodes. The top over here, IEA, inferred from electronic annotation. These are typically from prediction pipelines, and these are not manually curated. The ones below it, though, in the two different shades of green, are manual evidence codes that we curators use. Now, you see that they're three-letter codes. These aren't the ecocodes, these are the Go evidence codes, but they do map to ecocodes that you can search for in the new QuickGO, and I'll go over that again later. So to give you an idea of what these actually mean, let's talk about, for example, IDA, inferred from direct assay. These would include enzyme assays, immunofluorescence, cell fractionation, and in vitro reconstitution or transcription. And then some of the ones that don't really are not referred from direct kind of experiments are things like traceable author statement, where we can read a paper and we see that a statement is referenced, and we can actually capture that in the Go if we have an equivalent Go term for it. So how do we view and access annotations? Well, here at EMBL-EBI, they developed a tool called QuickGO, which pretty much allows you to browse the ontology as well as all the annotations available in the Go database. Now, this is the web link for it, and if you click there, it will take you to the actual website for QuickGO, and you'll see this format. But as being part of this webinar, we're here to actually talk about the new version of QuickGO that we will be launching tomorrow. So you will not see it just yet on the website, but if you go to that link, you'll see at the top of the screen a link to the beta. So if you want to follow along and click along, you can do that now. So I'm very happy to show you now the new version of QuickGO. So as you see, it's quite updated. It's very modern looking compared to the other one, so we're quite happy about that. And as of tomorrow, the web address will still be QuickGO, but we've actually improved what you can do with it compared to the other browser. And I'm going to walk you through the steps right now of the different things that we can do with it that you weren't able to do previously. So let's start with search. So obviously, you see the search bar. I've highlighted here in red with a red circle. So we're going to talk about searching. With the new search function, you can search for multiple types of gene product ontologies. So that means you can now search for proteins, protein complexes, RNA, Go terms, and ecocodes. So you can use uniprot identifiers to capture, to find your protein of interest. Protein complex identifiers are from Complex Portal. And RNA, you can use RNA central identifiers. Go terms remain the same as before. And ecocodes, if you have a particular favorite ecocode, you can also search for that. Keyword searches are also still supported. So if you don't know your Go term or ecocode ID, you can simply just do a keyword search. And to give you an example of this, we're going to go ahead and do an example search for the Go term apoptotic process. If you were to type that in the search bar and hit enter, you would actually end up with a screen like this, where it shows you two things, terms and gene products. In this case, in the terms, we see all the different terms that are related to apoptotic process, and what it shows you next to it in blue is the number of annotations made to a Go term and its descendants. So when you see apoptotic process, we see that we have 49,210 annotations. That doesn't mean we have 49,210 annotations to the term apoptotic process. This also includes its descendants, which are listed over here, these children terms. And the same thing with gene product. This is part of the keyword search, so it finds any protein that may have apoptotic process in the name, and you'll also see the number of annotations made to that gene product. So you can click on those and just view them. In this case, we're going to be clicking on one of the child terms. Here, we're going to be clicking on inflammatory cell apoptotic process, which has 155 annotations, to view what a Go term page looks like. So here we are. This is our Go term page. As we see at the top, we have our Go term ID, and then underneath what it's mapped to, so inflammatory cell apoptotic process. Next to the Go term ID, we see a pink P with a circle around it. This actually refers to the aspect of the ontology it refers to. So P refers to the process. If you see one with an F, it refers to molecular function. And if you see one with a C, it's cellular component. Next to it, you see a little basket. If you click on that, it will add it into your basket, over the Go term to your basket. And I'll go over why that's kind of important later. You can see now also underneath this, it says biological process, followed by the definition. And underneath that, if you click on that button, this says 155 annotations. It will automatically filter the Go database and allow you to view all the annotations to this term. Now, I'd also like to highlight for you guys over here on the right-hand side again is a new navigation bar that allows you to browse the different sections of the Go definition page. So you can look at things like the synonyms, the ancestry chart, so if you want to see where this term falls in the ontology and what it's related to, as well as the child terms of it. You can also look at the guidelines for how we would use this Go term in annotation, the taxon restraints for it, so what taxons it can be applied to and which ones it cannot. This is combined, the taxon constraints are combined with the annotation blacklists. And then co-occurring terms, so are there cases where you see this Go term, what are the other common terms that are usually annotated along with it? And then finally, if you're interested in how the ontology is developed, because it's constantly changing and edits are always being made to it, we've included the changelog where you can actually track how the term has changed over time and how it's been redefined or new synonyms have been added or a new type of relationship has been removed or added. So now that you've seen what a Go term page and you understand a little bit about how the search works, let's talk about actually looking at all the annotations that are in the Go database and how to filter them for the ones you're interested in. So to do this, we go back to our front page. We're going to go ahead and click on the View Go Annotations. And it's going to bring you to a screen like this, where it shows all the annotations in this really nice table displayed down here. And I'm going to talk to you about the filters right now. So we have filter. You can filter by several different things. We can filter by taxon ID. So if you only want to look at, let's say, things from the human proteome, so that'd be taxon 9606. And if you don't know the taxon ID for your favorite species, if you click on that button, it will bring a drop-down menu, and it has a link to the taxonomy page on Uniprot where you can search for your favorite species. Next to it, we have the gene product ID. So if you already know the identifier of the protein you're looking for, or you have several of them that you want to look at once and see all the annotations to them, you can put them in here and actually filter for them. Next is the gene product type. The gene product type is now referring to more whether you're interested in looking at proteins, protein complexes, and RNAs. So you would just select whichever, just the filter for, let's say, all the annotations you have to RNAs. You can do that there. Next is Go term. If you're looking for a very particular Go term and see what proteins have been annotated to it. So if you're interested in, let's say, using the example of apoptotic process, all proteins involved in apoptotic process, you'd grab the Go term ID and pop it in here, and you would be able to filter for all the annotations to that particular Go term. The Go aspect, so if you want to only look at cellular component terms, you can filter them there. The evidence type, whether you want electronic annotations or a particular type of manual annotation, you can look for them there. You can also pop in an eco code there if you're looking for a particular experimental type. And then finally, the annotation extension. These are what I mentioned before. So if you're interested in searching for, let's say, everything that occurs in all the annotations that are available for epithelial cells, you can search for the URON code there. Next, you have the More button, which brings you to this little pop-down menu here. And you see that we have the qualifier terms. So if you're interested in looking at a particular qualifier, you can search for it there and select it. A reference, so if you're interested in a particular source, let's say you only want to look at things from the unirule pipeline that automatically generates Go annotations, you can search for it there. Or if you want to search by PMID, you can do it there, and you can actually type in the PMID you're interested in to see if it's actually been annotated already in the Go database by selecting PMID and then typing it in the Add More. With From, if there's a particular product if you're interested in a gene product that has interactions with let's say something from the QuickGO database, so a drug of interest, or another Uniprot identifier, you can filter it from there. And then, of course, if you want to only look at annotations from a particular database, you can always sort by assign by. Going back to our annotation table screen, we have two more buttons that I didn't mention before, which are the Customize and Export buttons. For these, you can add or remove columns using the Customize button. So this will actually impact what you export as a tab delimited file, which is what the Export button will allow you to do. So you can export all the filtered annotations you want, or the entire, well, I don't recommend the entire data set because it is quite large, but you can filter for the ones you're interested in and export them as a GPAD file, a GAF file, or customized tab delimited file. The tab delimited file will actually only show with the columns that you've selected for or you've added on. Now, you're probably wondering, do I have to, let's say I want to filter for a Go term, do I have to go back to the Go term page to actually see if this is within the right branch of the ontology that I'm interested in? And the answer is no. You can actually view the Go ancestry tree by clicking on this little button over here. It looks a little bit like a tree, so we're just using the example of catalytic activity. And if we click on that, it brings us to the ancestry tree. I know it's not catalytic activity here, it's actually a type of binding. I felt this was actually a bit more interesting, so it showed a little bit more in the ontology and the depth that you can have to it. The other thing that's actually quite unique for our browser is that you can now actually view the ecocode ancestry tree as well, so you can understand where evidence codes come from. So if you click on that, in this case, I'm using the example from annotations from CollectTF, which used an EMSA assay to detect how a protein binds a particular piece of DNA, and you can see how that's related to the different types of evidence codes used, and the ecocodes are on top of it. So you can actually search for these ecocodes to kind of better understand the evidence ontology, which is something that wasn't available in the previous QuickGO. Now, we're going to go on to something that's a little bit different than the other two, and this is a little bit specialized, but it's actually quite useful, especially when you get more and more into gene ontology and gene ontology annotation, and that's creating Go slims. So going back to our front page to investigate this, we're going to go and click on Explore Biology. But before we actually go and set up a Go slim, let's talk about what they are and why they're useful. Go slims are cut-down versions of the Go ontologies containing a subset of the terms in the whole Go. So there's about 40,000 Go terms, and you're not going to want to view all of them. You're just going to want to look at some of them because not everything is going to be of interest to you unless, of course, you're doing whole proteomic screening. So what do? They give a broad overview of the ontology content without the detail of the specific fine-grained terms. So these really specific concepts, this is just going to give you a general overview. And they're really useful when you're trying to get a summary of the results of, let's say, a genome or microarray or cDNA collection. And you can just get kind of a broad classification of the gene product function that you want. And you can create these yourself, or you can actually use the predefined sets that are available from the Go consortium, and these are already programmed into QuickGO. So going back to the Go Slim page, so if you clicked on Explore Biology, it'll bring you to a page that looks like this. Now, you can select from the predefined Go Slims by clicking on Select. In this case, I've chosen the viral process Go Slim, the viral Go Slim, and it's 29 biological process terms. They're listed down here. If you wanted to create your own Slim, you can input your own Go terms. So if you have them written down on a piece of paper or in an Excel file, you can copy and paste them into there and add in your own individual terms. And then if you hadn't put some Go terms in your basket, you can select from your basket and add them. In addition to this, you can actually filter for gene product ID. So if you're only interested in a few proteins and see what annotations they fall within, let's say in this case the viral Slim, you can do that way. And you can also restrict by taxonomy. So if you only want to look at a particular species of virus, you can do that here as well. So after you've done all this, you can also deselect terms that you don't want to view here by clicking on the X. After that, hit Apply. And it brings you to a page that looks like this. This is, again, the annotation table, but it's slightly different now. So what you'll notice is that we have two columns, Go term and original Go term. Original Go term is actually the term that you slimmed for. The Go term is all the terms that actually map to this Go term. And if you click on the ancestry tree, you'll actually see from the original Go term how these other ones are related. And you'll see, so it pulls out all the annotations that are mapped to that Go term in the Slim. The other thing that's quite useful about this, if you just want to get a quick summary of what's there, you can click on the Statistics tab. So when you go in there, you actually get to see the report that tells you how many annotations and gene products map to your Slim. And you can also see how many unique Go terms there are, which Go aspect is best represented. So in the viral Go Slim, since it's all biological process, you'll only see that the aspect is biological process. The types of evidence that were used, whether they're electronic annotations or manual annotations that curators have created, the species that are represented, and also the database that assigned them. Now, in this case, so that pretty much sums it up for Go Slims. It's a very complicated topic, and if you guys are interested, please provide some feedback, and we'd be happy to try to arrange a webinar on that. But I'm going to talk about the last part of the usefulness of our QuickGO page, and that's the basket. If you've been adding Go terms in your basket, and they're all from different parts of the ontology, you can actually find a way to compare them there, or you can use the basket to create custom ghost limbs as you browse the ontology and added them in. So this is what our basket looks like when it's empty. So ahead of this, I did a quick search for TGF-beta, and I've added in some of the Go terms that are available there and within that branch of the ontology. So within the basket, you see that we have several options. We can view the ancestry tree, which we'll do in a minute. We can filter with the terms. So if you click this button, it'll take all the Go terms in your basket and filter the Goa data set. And so you'll only be able to see annotations to these Go terms. You can clear it, so if you want to empty it, and you can export, which will then export all the annotations made to these 4 terms. In addition to this, you can add terms here if you know the Go ID, but of course you can always just add them as you did it when you were browsing. So if you wanted to understand how all of these annotations were related, all these Go terms are related to each other, you can click on the ancestry tree icon here, and it brings you a nice ancestry tree chart over here that actually shows you how transforming growth factor beta receptor signaling is related to all of these processes. And of course, we're always looking for feedback, and help is always available to you if there's something that wasn't quite clear. You can always visit the FAQ page to get a general question answered, and if that doesn't answer your question, you can always click contact and get in touch with us directly. And on top of that, since this is a new version of QuickGO and we are trying to get user community feedback, please let us know what we're doing right and what we can improve. There's always room for improvement, so if you see something is not up to your specifications that may help your work, others most likely will want it, and you can email us and we'll try to see what we can do about it. Now, saying that, we do have some planned improvements for QuickGO already. There's going to be the enhancements to filtering extensions, more interactive ancestry trees, which will allow you to kind of customize what you see in the ancestry tree, and then ID mapping to other sources. ID mapping was available in the old QuickGO, and the old QuickGO will be up and running still. It will just have a link from the new QuickGO page, and from there, with ID mapping, what you'll be able to do is just take, let's say, a UniProt identifier and map it to other databases of your choice. So let's say if you have a mouse protein in UniProt and you want to see it on the MGI database, ID mapping will give you the MGI accession ID. With that in mind, I'd like to thank you all for listening. Feel free to contact us with any questions or comments via the website or our email. The website, as listed before, is www.ebi.ac.uk/QuickGO, and our email is goa@ebi.ac.uk. Now, this is the last webinar for right now until October, but please keep an eye for upcoming webinars in October, so at the page here for training. And of course, please provide any feedback. Thank you very much.